Hello friends, welcome to IFAS. Today we are starting a very simple topic of biochemistry. In this lecture, we are first going to study what we mean by atom, then what are molecules, and we will try to gain some information about the bonds. So, are you ready? What you have to do is, you have to listen carefully. While hearing my lecture, if you find some point very important, note it down. Means if there is something new for you, please note it down. Again, every word what I said is not important, or what I want to say is that it's not necessary that you write every word in your notebook. Try to make short notes, listen everything, try to make concepts, and wherever you find that this line is important and in the near future you can forget it, just note it down. For making notes, you can take help of these teacher handouts. Whatever I am writing, you can take help out of this. Okay, so hopefully you will enjoy this lecture. So let's start with a very simple question, that what is matter? Matter could be defined in multiple ways. The simplest way of defining a matter is anything which can be physically sensed, which we can touch, which is physically existing in a nature, is a matter. In other words, the pen by which you're writing is a matter. You yourself is also a matter. I'm also matter. This room is matter. So anything which is having its physical existence would be referred to as matter. Now, there may be various states of a matter. That is, whatever physically exists, it may occur either in a solid form, or in a liquid form, or in a gas form, or in a plasma form. That is, matter could be in a solid form. For example, the calcium carbonate salt, or even the pen which you are holding in a hand, is a matter in a solid form. The matter could be in a liquid state. The best example is water. What we drink is in a liquid state. The gases include oxygen, nitrogen, carbon dioxide, etc. These are also matter which are having a physical existence. And when these gases they are superheated, they can move to a fourth state. And that fourth state of matter is referred to as plasma. So we can simply say that plasma is a superheated state of a matter. Basically, whether a matter is in a solid state, or a matter is in a liquid state, or a matter is in a gaseous state, or a matter is maybe in a plasma state, the basic composition of any matter is an element. Okay, so what we say that matter is made of combination of elements. Okay, so matter, it is made of combination of an elements. But now, dear friends, I know there is a question in your mind that what is an element? We can define element in multiple ways. The simplest definition of an element may be that element is a substance that cannot be separated into a simpler substances by a chemical means. Okay, so we can write it down that an element is a substance basically which is having the substance. There are various atoms that cannot be separated, into simpler substances by chemical means. Here what I want to say is that, let's say we are talking about an carbon element. So a carbon element, it will be composed of a similar kind of atoms. You cannot get any other kind of atom in that particular element. In other words, we can also say that an element is basically a pure combinations of an atom, because matter, it is composed of various elements. Matter may be composed of silica, an element, iron, copper, nitrogen, phosphorus, sulfur, magnesium, etc. And each element which is composed of similar kind of atom, it cannot be separated into another simpler means. Okay. In other words, we can say that basic unit of this element is an atom, or we can say repeating unit is an atom. Are you getting this? So what we have said up to now, that everything which is around us, whether it is a solid, liquid, gas or plasma, is composed of a matter. Matter is a heterogeneous mixture of various elements. Element is a pure combinations of homogeneous atoms or similar kind of atoms. And what is an atom? If someone asks us what is an atom, so we can define like this, that atom is the smallest particle of an atom, which retains all the identities, or we can say chemical properties of an element. Okay. So if let's say there is some carbon element, the repeating unit which is present in this carbon element is an carbon atom, and whatever the properties which are shown by this carbon elements, these all properties, they are also shown by this carbon atom. So we can say that the repeating unit, or we can say basic unit, which is present in a element is referred to as an atom. Okay. So what from this first slide, what should be clear, that what is matter, what is element, and what is an atom. So let's move to next slide. Now question may come in your mind, that how many elements are known. As we have already discussed, that every element is composed of atom. In other words, what I am asking is, how many different kinds of an atom they are present in nature? Any guess? Yes. Just think about a periodic table, what you have studied in chemistry in your mind. You will start getting the answer. Yes, you are true. So friends, we know that there are over 115 elements. There are over 115 elements known to us. So for various elements, hydrogen, helium, lithium, boron, carbon, nitrogen, oxygen, fluorine, neon, these all are elements. Sodium, magnesium, aluminum, silicon, phosphorus, sulfur, chlorine, argon. Means you can completely memorize and think about a periodic table. Out of these 115 elements which are known to us, dear friends, approximately around 90 elements are naturally known. Means you can have a look on a periodic table. This is a periodic table. In this periodic table, there are, although various other elements they have been added, over 115 elements they have been known, but only around 90 are naturally known. Rest are artificially produced. Various elements, that is approximately 30, have been produced in lab conditions. Okay, so almost 90 elements, and repeating units in an element is an atom, so we can say around 90 elements or in atoms, they are naturally known out of 120. Rest of the atoms or elements, they are produced by nuclear fusion process and by bombardment of gamma particles, by bombardment of positron particles. We don't need to go in that much depth. So almost around 120 elements, out of which 90 they are naturally known. And friends, if you think about that, out of these natural 90 known elements, how many elements they are present naturally in biological system? Are all elements which are present in nature, they become a part of biological system? No. Answer is no. That is, we or a plant or a microbe or a fungus is not composed of 90 different type of elements. If we think about biological organism, what you'll find that approximately there are, again I am saying approximately, there are 25 elements. Yeah, we can say only 25 elements are present in biological system. Only 25 they are present in biological system. So everything which is present in a nature is not a part of biological system. And when we are talking about these 25 elements, we are talking about all macro as well as micronutrients. We are including every possible type of atom which could be present in biological system. My dear friends, this is a list of our major atoms present in biological system. What you find over here is that this distribution is according to fresh weight. So according to fresh weight, the maximum atom by weight is an oxygen, followed by carbon, then hydrogen, nitrogen, calcium, phosphorus, potassium, sulfur, sodium, chlorine, magnesium, and then there are various approximately 15 trace elements which include boron, chromium, cobalt, ferrous, iron, zinc, molybdenum, vanadium, etc. So the atom which is present in maximum proportion is by weight is oxygen. So atom by weight is oxygen. and atom by occurrence. That is, which atom is maximally present. If you think like this, then you'll find it is a hydrogen atom. Hydrogen atom, although its contribution from weight point of view is lesser, but it is much abundant even than the oxygen. The reason is straightforward. Whether you talk about water, H2O, it is having 2 hydrogen and only 1 oxygen. Even if you talk about carbohydrate, C6H12O6, oxygen is 6 and hydrogen is 12. So if you talk about the number, hydrogen will top the list. But if you're talking about the weight, oxygen is the most, we can say, common atom or contributing maximum to weight to any biological system. And keep this thing point in your mind that we are talking about a fresh weight. So atom, fresh weight, because if we dehydrate the material and then we try to find out that which atom is contributing maximally, then it will come out to be carbon. So dry weight, then the maximum contribution is by carbon. So again, these 3 things, they should be kept in a mind. Most abundant atom, hydrogen. Maximum contribution in a weight on a fresh weight basis, oxygen. Maximum contribution to a weight if our biological sample is dry, then it is carbon. Okay, so let's move to the next slide. That if we look carefully on all these elements, again, all these elements are not macro nutrients or macro elements. All these elements, they are not so abundant. Only out of these 25 or 26, 6 elements, they are very abundant in biological system, or almost we can say that all biomolecules, what we see in biological system, they revolve around these 6 elements or atoms. So these 6 elements are carbon, hydrogen, nitrogen, oxygen, phosphorus and sulfur. That is, if you talk about the carbohydrates, what you'll find is, if you talk about carbohydrate, it is composed of carbon, hydrogen and oxygen. If you talk about proteins, which are made of amino acids, you will find that there is carbon, hydrogen, oxygen, nitrogen and one more atom may be there. Beside nitrogen is sulfur. If you talk about the nucleic acids, that is, if you talk about DNA or RNA, what you find is they are composed of carbon, hydrogen, oxygen, nitrogen, and instead of sulfur, that would be a phosphorus. That's why often sulfur is utilized for labeling of proteins, while the phosphorus, radioactive phosphorus, is utilized for labeling of nucleic acids, because phosphorus is not present in protein and sulfur is not present in nucleic acids. And the 4th biomolecule is lipid, which is again composed of carbon, hydrogen and oxygen. So you can see that there are only 6 elements, that is carbon, hydrogen, oxygen, nitrogen, phosphorus and sulfur. These are the 6 elements. which are abundant in biological system. If you see the position of these 6 elements on periodic table, what you will find is that they are present either in period 1, 2 or 3. For example, here you can find a hydrogen in period 1, carbon, nitrogen and oxygen in a period 2, phosphorus and sulfur in the period 3. You are not having the elements belonging to period 4 or period 5 in a biological system. And as you may have a little bit idea about the distribution of an elements in periodic table, that as we move from top to bottom across periods, the size of an atom increases. That is, these are of small size and these atoms, these would be of large size. So what we want to say is that the atoms which are of larger size, which are of bigger size, they are not seen in biological system. Carbon, hydrogen, oxygen, all they are of small size. Even nitrogen is also small, and phosphorus and sulfur, they are also relatively small because they belong to a period 3. Now the question, in other words, what we want to say is, from here the point to be note down is that atoms of small size, atoms of small size are preferred in biological system. The atoms of small size, of small atomic radius, they have been preferred in biological system. Now a question may come in your mind that what is problem with large size atoms? Why large size atoms like gold, like iron, like alumina, like magnesium, like uranium, thorium, rubidium, they are not preferred? What is the reason? Because if you will have a large size atom, you will need a lesser number of atoms. And if you are using a small size of atoms, you will have to use large number of atoms. Why these large size atoms, they are not preferred? Why the atoms with a small size, they have been preferred in biological system? The answer is straightforward and very simple. That basically the simple concept is that atoms with small size forms stronger bonds, and stronger bonds means more stability. We do not need a biological form which is fragile. We do not need a protein which can easily get lysed into a constituent amino acid. We need a stable molecular system, and for this we need a stronger bonds. Now coming to the point, why the atoms with a small size forms a stronger bonds? Any idea? Any guess? Yes, you are correct. As the size reduces, the surface area to volume ratio increases. And if surface area to volume ratio increases, there is more overlapping of surfaces because more surface area is available as compared to volume. More surface area come into contact or more surface area overlap means more stronger would be bonding. So what I want to say is that small size atoms, they form stronger bonds because as size decreases, surface area to volume ratio increases. Are you getting this? Surface area to volume ratio increases. And increase in surface area as compared to volume. This increase in surface area as compared to volume allow atoms to come in contact better, more stably as compared to an atom where a very small surface area is in contact as compared to the volume. Just think about a scenario. The two small balls, they are in contact and you can say glued them through a Fevikwik. Two small balls and you have connected them through a Fevistick. And the second time, now what you're taking is two large balls and again using a same amount of Fevisticks and try to join them. The surface they are coming, wherever the surface they are coming in contact, you are putting a Fevistick and try to ligate them. Which of them would be more stable? The two small bonds glued through Fevistick or two larger balls glued through a Fevistick? Definitely, you know, answer is that the small balls, if they have been fixed with a Fevistick, the system would be comparatively more stable because here the surface area to volume ratio means as compared to volume, the surface area which is coming in contact is comparatively larger, and that's why the bonds are stronger. Then another benefit of having the small size atom is that there is flexibility. These small size atoms, they can flexible to form 1, 2, 3 or even 4 covalent bonds. For example, you can see carbon is forming a 4 covalent bonds. So second important thing what you can note down is flexibility of forming 1, 2, 3 or even 4 electron bonds or covalent bonds. That is another benefit. The larger size atoms, they do not have this much flexibility. Carbon can form atoms with 4 other atoms, similar kind or even dissimilar kind. And they have a capability to form multiple type of bonds. That is, we'll study that these atoms, they would be involved in, no doubt, covalent bonds. And beside covalent bonds, in covalent bonds, they can form a single covalent bond, double covalent bond, even triple covalent bond. Beside covalent bonds, you can find them to be involved in a hydrogen bonding, electrostatic interactions, van der Waals interactions, hydrophobic interactions. So this is another reason. So 3 points you have to note it down, that why these small size atoms, they like carbon, they are preferred in biological system. First, they form a stronger bonds. Second is there is a versatility in the bonds, or we can say flexibility in the type of bonds. And third is capability of formation of multiple type of bonds. Okay, we noted this. Okay, good. Now If you are not able to note it down, note it just down. Now, moving to next. So, moving to the next slide. That is, now, question may come in your mind that why life is carbon based, not silicon based. Life is carbon based. There are multiple reasons. First, carbon being a half filled, means in its outermost shell of an carbon, what you will find is there are 4 electrons. So this is nucleus, this is an inner orbital which is having 2 electrons, and outermost shell is having 4 electrons. It is half filled. So it can lose or gain 4 electrons, or it can form, you can say, bonds with 4 other atoms. Means carbon can form 4 covalent bonds with carbon or other atoms like hydrogen, oxygen, nitrogen, phosphorus, sulfur. Carbon can form a bond with a carbon. That is very important for making a polymers. This concatameric property is very important. A stable concatamers, they can be formed if we are using a carbon. The atoms like nitrogen, even if they form a bond, they form concatenarize, that is unstable, very unstable. Even boron will not form a stable concatameric form. If you need a polymers like proteins, if you need a polymers like carbohydrates, you know, or even simply hydrocarbons where you are having bonding between carbon. These are very stable. The bonds which would be formed by silica with a silica would be very weak, fragile. So silica cannot be utilized. The silica bond, it is very unstable. Especially in water, in presence of aqueous environment. So even silica forming its oxide with another silica, these polymers are possible, like methylated silica. These polymers are possible, but again these would be unstable in presence of water. So like carbon, silica belonging to a same group may form a 4 bonds, but the problem is the silica concatamers are very unstable. The silica oxygen bonds, although such polymeric structure or concatenaric structures, they can be formed, but again they are very unstable in biological system as compared to the bonds. For example, in 2 carbohydrates, here you can also find a carbon oxygen bond, but this is comparatively or relatively stable as compared to silica oxygen bond. So homomeric carbon concatameric form, such bonds, they are very stable as compared to homomeric nitrogen or boron or even silica. Carbon can form a heteromeric concatameric structures, that is carbon oxygen. Silica can also do so, but such structures, they are very unstable, and especially in the aqueous environment. So silica based life can be possible only in a scientific, yes, you can say scientific fantasy films. In most of Hollywood movies, you can find that there are some aliens whose structure is composed of silica or silicates. But practically it is not possible on Earth, because everywhere there is a water. In presence of water, any structure which would be of silica or silica with the oxygen would be very unstable. You getting this point? Okay. Now let's move to the next problem. That why elemental composition of a biological system do not match with the elemental composition of earth crust where we live. The earth crust where we live, it is mainly composed of silica. You can find that it is mainly composed of silica, aluminum, iron, calcium, sodium, oxygen, potassium, magnesium. While the biological system is composed of mainly carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. These kind of atoms, they are not present maximally in biological system. Why there is a mismatch? The answer is straightforward and very simple. That the elemental composition of a biological system do not match with an elemental composition of earth crust, because the origin of an early life form was not on earth crust. The origin of an early life form was somewhere in a water body. Okay, so answer to this question is origin and early evolution up to even Cambrian period. Origin and an early evolution was aquatic. So origin and early evolution of life was aquatic. So whether it is in formation of molecules or macromolecules or some kind of protobionts or fossil life forms or further variety of other forms, this whole early series of events, they have occurred in water body. Some primitive water pool as suggested by Oper and Haldane, what we generally study when we study the origin of life. So that's why the elemental composition of the biological system matches more with a aquatic system as compared to the terrestrial or the earth crust where we live. You got this point? So like a water, we have more amount of hydrogen, we are having more amount of oxygen. The amount of carbon is also relatively more in a water as compared to an earth crust. That's why we find more carbon. Okay, even the pH of biological system, the salt composition of biological system, etc., they also resembles more like a sea. Okay, so you got, I think probably you got the answer that why the biological life form elemental composition resembles more with an aquatic system as compared to the earth crust. Okay, so this was the beginning of this topic, that what is matter, how many elements they are known, what kind of elements they are present in biological system, why small size elements they are being preferred in biological system, what is problem with the silica, then why the elemental composition do not match with an elemental composition of earth crust. So this was an introduction to the world of biochemistry. Then let's move to a little bit chemistry part, that what actually we mean by an atom. So let's focus on the structure of an atom. My dear friends, atom, which is we know and we have already defined, is the basic unit of an element. It has a nucleus. In atom, there is centrally situated nucleus. Nucleus has two kind of particles, namely protons and neutrons. And you know very well about this, so I'm not going to much detail. Since the proton bear a positive charge and neutron do not bear any charge, so nucleus is a positively charged. So nucleus is positively charged particle. Around the positively charged nucleus, there are certain shells or electron shells, and in these electron shells, the electrons orbit around the positively charged nucleus. Okay, so there are three kind of subatomic particles. In a nucleus, you are getting a proton and neutron. The nucleus is positively charged. That's why negatively charged particles, they keep on revolving around this nucleus, and these negatively charged particles which are revolving around nucleus are referred as electrons. Okay, I know you are very much aware of this thing. Now let's have a look about the mass and charge of these subatomic particles. Just note it down. The mass of a proton is 1.67 into 10 raised to minus 27 kilogram, or you can also say that it is 1.67 into 10 raised to minus 24 gram. Charge of the proton, generally we say that it is a +1, but what we mean by +1? It +1 means 1.6 into 10 to the power minus 19 coulomb. 1.6 into 10 to the power minus 19 coulomb. This is the charge. The mass of neutron, my dear friends, is almost similar to the mass of a proton. That is, again, it is 1.67. 1.67, although little bit, if you analyze critically, it was 1.6726 and it is 1.6749, negligibly higher. So it is 1.67 into 10 to the power 27 kilograms. And as name suggests, neutrons, they are neutral, that is, they do not bear any charge. So mass of neutron is similar to the mass of proton. But the charge of proton and charge of neutron is different. Charge of proton and charge of electron is equal, except that they bear a opposite sign. That is, charge of the electron is -1, and what we mean by -1? Again, 1.6022 into 10 to the power minus 19 coulomb. While the mass of an electron is negligible as compared to the mass of proton or as compared to mass of neutron, it is only 9.1 into 10 to the power minus 31 kilogram, or if you convert into gram, it will come out to 9.1 into 10 to the power minus 27 kilogram, or more totally 28 kilogram. So here, if you talk about the mass of an electron, you can say that mass of electron is almost 1840 times lesser as compared to mass of protons or neutrons. So in other words, we can say that mass of an electron is negligible. when there would be 1840 electrons, the mass of this would be equivalent to 1 proton. And we know that this scenario is never going to happen, because even if you talk about the atom with an atomic number 120, it will have 120 electrons, and the mass of 120 electrons would be even far lesser than the mass of 1 protons. So in other words, since the mass of the electron is negligible as compared to the mass of proton or neutron, we say that the mass of an atom resides in nucleus. It is due to the protons or it is due to the neutrons. Okay, so mass of nucleus resides in nucleus, and from nucleus we mean that it is due to proton and neutron. Are you getting this? Why we are not including the mass of electron? The mass of electron is negligible as compared to the mass of proton and mass of neutrons. Okay, you got this point now. Let's move to another point. And whenever we are talking about the charge, so instead of mass of nucleus, mass of atom resides in nucleus, and charge of atom depends on the number of protons and electrons. It depends upon protons and electrons. Okay, got this point? Okay, let's move to another part. That is, what we mean by atomic number. Up to now it is clear now that, and probably you would have also noted down what is mass of an proton, charge of a proton, mass of electron, and so on. Then let's define what is atomic number. Now friends, atomic number basically it is an identity of an atom. It is an identity of atom. And it depends upon the number of protons in a nucleus. That is, an atom which has a single proton in its nucleus, its atomic number would be designated as 1. So, for hydrogen, the atomic number is 1. For the oxygen, it is 8. For nitrogen, it is 7. Why? For carbon, it is 6. So these are the atomic number. So what is the factor on which this atomic number depend? So atomic number depends upon the number of, protons. Okay, you're getting this point? So why we consider why we consider only the number of protons? Why not electrons? Why not neutrons? Any thought, any idea about that? So, got the answer or not? Hydrogen, oxygen, nitrogen, or any other atom, it is identified by the number of protons. Why? Why we do not rely on number of neutrons, or why we do not rely on number of electrons? The reason is straightforward. The number of neutrons may vary in an atom without altering its chemical properties, as we will see later on in the isotopes. Carbon 12 form will have a 6 neutron, carbon 13 will have a 7 neutron, and carbon 14 will have a 8 neutrons. So, what would be the identity of carbon if you are giving it on basis of the neutron 6, 7 or 8? And regardless of the number of neutron, whether it is 6 or whether it is 7 or whether it's 8, the properties, the chemical properties of atom is same. It is a carbon atom. So we cannot rely on the number of neutrons. And we will also see later on that atom can lose an electron and atom can gain an electron. The number of electrons may also vary. So even we cannot rely on the number of electrons. We rely mainly on the number of protons, because number of proton is a static factor. If this factor varies, then the identity of an atom will also vary. Then we will say that now it is not a carbon atom. If the number of protons instead of 6, it becomes 7, we will say that now it is not a carbon atom. It is showing a properties like a nitrogen. And or if protons become 8, we will say that no, now it is not nitrogen. Its chemical behavior is like a oxygen. Okay, so getting this point? So, in other simplest word, the atomic number is based upon the number of protons. Okay, then these atoms in a modern form of periodic table, if you see them, they have been arranged according to the atomic number. For example, first there is a hydrogen, then there is a helium, lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine. What is the basis of the distribution? That its atomic number is 1, its atomic number is 2, its atomic number is 3, this is 4, this is 5, this is 6, 7, 8, 9, 10, sodium is 11, magnesium is 12, and so on. Aluminum is 13, this is 14, silicon, this is 15, 16, 17, 18, and so on. The arrangement of atoms in a periodic table is based on number of protons. Okay. Then moving further, that what we mean by atomic mass. As I have already mentioned, the atomic mass, it resides in nucleus. It resides in nucleus. In other words, we want to say that this atomic mass is due to mass of protons plus mass of neutrons which are present in the nucleus. Although we know that in an atom beside proton and neutrons there are also electrons, but we do not consider the electron in a mass, atomic mass calculations. The reason, as I already mentioned, the mass of electron is negligible as compared to mass of proton or mass of neutron, so that mass of electron can be easily neglected. So mass of electron is negligible as compared to proton or neutron, so it do not contribute to atomic mass. It is not contributing much to an atomic mass. For example, if we have to find out the mass of atomic mass of carbon, how you will find atomic mass? What is the atomic mass of carbon? How you will find? Then you must know that what are the number of protons and what are the number of neutrons in carbon. Find that carbon has 6 proton and 6 neutron, so atomic mass will come out to be 12. Okay, are you getting this point? So carbon has 6 protons and 6 neutrons. So atomic mass comes out to be 12. Similarly, if someone asks you what is the atomic mass of oxygen, how will you find atomic mass of oxygen? So again, yes, what is your answer? 16. Again, it's not necessary that it would be always 16. The thing is that no doubt it depends upon proton as well as number of neutron, and generally the number of proton and the number of neutron both are 8 and 8, then it would be 16. But you know that there may be an isotopic form where the number of neutron could be 9. Then the mass of same oxygen atom would be 17, and there is one form of oxygen atom where the number of neutron is even 10, then it would be mass of an oxygen atom could be 18. So atomic number is fixed. Atomic mass of an atom is a variable parameter. That is, atomic mass depends upon the number of protons and the number of neutrons. And since the number of neutron varies, the atomic mass can also vary for any given atom. Only one thing which you have to keep here in mind is that atomic mass is the number of proton and the number of neutron. Then let's move further. What are isotopes? A question may come in your mind that what are isotopes? Just make any kind of guess. What are isotopes? Yes. So isotopes, as name suggests, that they are same form of atom. They are, we can say, so it is different form of same atom. Okay, it is a different form of same atom. So atom is same. It is different form of same atom that occupy same position in periodic table, because if it is the same atom, it will occupy the same place. So iso means same, top means position. So they are occupying a same position in the periodic table. Now coming to the points, why they are occupying the same position in periodic table? Because we know that modern form of periodic table, it has been arranged according to atomic number. So what we can say that isotopes, they have same atomic number. They have same atomic number. They have same atomic number means, it means they will have same number of, they will have a same number of protons. But then why we are saying that they are the different forms of a same atom? At least there must be some criteria, or there must be some parameter which must be variable. That is, they have variable number of, yes, neutrons. They have same number of protons, but they have a variable number of neutrons. And we know that atomic mass depend upon the number of neutrons as well as number of protons. And since there is a variation in number of neutrons, there would be also a variation in atomic mass. Yes. So they have same atomic number, same number of protons, but they have variable number of neutrons. It means variation in atomic mass. So, and this variation in atomic mass is basically due to the variation in the number of neutrons. Yes. For example, we can take a case that these are the three different isotopes of hydrogen. You can see that these three forms of hydrogen include protium, deuterium, and tritium. You can see that the protium, that is normal hydrogen, it has 1 proton and 0 neutron. Another form of hydrogen, that is deuterium, which has 1 proton and 1 neutron. And third is a tritium, another form of hydrogen, which again has 1 proton but 2 neutron. This would be referred to as tritium. Are you getting this? Just note it down. Okay, so you noted it down. These are the three different forms of hydrogen atom. Then let's move further. We have considered about the atomic mass. We have considered about the atomic number. One more term which often you see in our literature is the atomic weight. So now a point may come in your mind that what actually this atomic weight is, because up to now most of students may think that atomic mass and atomic weight is a synonym, that it is one and same thing, but it's not like that. It is exactly not like that. The atomic mass of a carbon is 12, but atomic weight of a carbon is 12.01. Now, from where did we get this 0.01? So, the thing is, my dear friends, we know that in a nature, there are more than one type of a form of an atom, which are referred to as isotopes. And whatever the sample what we are going to collect from a nature, it will not have only one form. Often it will have, or it would be a mixture of the isotopes, and the composition of this, or we can say relative composition of these isotopes, would be as per their occurrence in a nature. The contribution in a weight is more due to those isotopes whose atomic mass is more. For example, if we try to collect a carbon from our nature, you find a carbon 12, which is almost 99% of the total carbon what we see in a nature, and there is a carbon 13, which is also present in nature, almost around 1%, although there is one more form of carbon that is referred to as carbon 14, but it is in a trace amount. It is so small that we are now not included that in our calculations. So whatever the carbon we are collecting, the weight of carbon would be 99% contributed by this 12. That is a carbon form which is having a number of proton 6 and number of neutron 6, that is a carbon 12 form. 99% of this will come into play. That may come out to be 11.88 as it being said. But we also, besides getting carbon 12, we are also getting a C13 form. We are also getting a C13 form, which is having 6 protons and 7 neutrons. So atomic mass is 13. So, but contribution of this is only 1%. 1% is 0.13. So total, or we can say average atomic mass would be combination of these two, which will come out to be 12.01, and this is referred to as atomic weight. So atomic mass, you do not get in a decimal, it depends on protons and neutrons. Well, atomic weight, it depends upon the contribution of each isotope which is present in that particular sample. So if the percentage of C13 increases, the atomic weight will gradually increase. In a nature, these isotopes, they are pertain in certain stable equilibrium ratio. Okay, you are getting this point? That what we mean by atomic mass and what we mean by atomic weight. Now, you would have heard a term radioisotopes. So let's try to understand what we mean by radioisotopes. Okay, so moving to another aspect, what we mean by radioisotopes. For example, if we talk about an hydrogen, we say that hydrogen has 3 forms: protium or normal hydrogen, deuterium, heavy isotope of hydrogen, and tritium. It is also a heavy isotope of hydrogen, but this is also referred to as radio. Tritium is referred to as radioisotopes. Similarly, carbon may have different form. There may be a carbon 12 form, a higher, heavier isotope C13 form, and further higher is C14. But the C14 is referred to as unstable form. So, what are radioisotopes? So, radioisotopes are isotopes which emits radiation in simplest word. And what we mean by radiations, that is which emitting alpha, beta, gamma, etc. These rays, they are being emitted, which can be easily measured. But why they are doing so? Why there are some isotopes? They are emitting radiations. The thing is that the isotopes has unstable, the isotopes have a unstable nucleus. What we mean by a unstable nucleus? We know that in a nucleus there are protons and there are neutrons. And if protons and neutrons, they are in equilibrium conditions, they are stable. If both are equal, the nucleus is stable. But whenever either protons over-dominate the neutrons or neutrons over-dominates the proton, there is a disequilibrium, which leads to a instability. So why unstable? Because there is a disequilibrium in number of protons and neutrons. There is a disequilibrium. Whenever it will happen, it will make a nucleus unstable. Any system, if it is unstable, it will always try to achieve stability. And same hold true for even these isotopes which are having an unstable nucleus. So what happens is, to achieve stability, these nucleus of these radioisotopes decay. So to achieve stability, we say nucleus disintegrate or decay and emits radiations. Because if something is decaying, that something is splitting, something is disintegrating, obviously something will come out of the nucleus. And why this is happening? The purpose is to achieve stability. So in other words, there is an emission of some radiations. So now we can define what we mean by radioisotopes. So radioisotopes are the isotopes or the forms of atom which have an unstable nucleus. These form of atom which are having an unstable nucleus, they try to achieve stability, and to do so, their nucleus decay or their nucleus disintegrate. And whenever a nucleus decay or disintegrate, it start emitting certain particles to the surrounding. So such atoms or forms of atoms which emits radiation to outside are referred to as radioisotopes. So all the forms will not be radioactive. And what is radioactivity? Radioactivity is an activity of the atom which is having an unstable nucleus. What kind of activity? That these atoms which are having unstable nucleus, they start emitting the radiations to outside. This activity is referred to as radioactivity. Are you getting this point? So you can easily see that in case of tritium hydrogen, there is a one proton and then there are two neutrons, so there is a disequilibrium. In C14 you can see that there are 6 protons and there are 8 neutrons, so again there is a disequilibrium condition, and purpose is simply to achieve stability. We will study these radioisotopes and radioactivity much detail in the radioactivity units. Whenever we study the radiations and radioactivity in detail, there we study more about these. But to simplify the things over here is that these are radiations or nuclear decay. They can emit 3 different kind of particles. These radioactive decays of 3 types. First, these 3 types are alpha rays, beta rays and gamma decay, although there are some other forms as well. In alpha decay, a alpha particle emits, and alpha particle which emit is similar to the nucleus of helium atom. And what nucleus of helium atom has? It has 2 protons and 2 neutrons. So since it is a nucleus, it would be positively charged. So that's why we'll see that alpha particles, they are positively charged. Whenever there is a beta decay, and in beta we will study later on, if in beta this is a negatron kind of decay, this beta particle is like an electron which bears only charge. It do not have any mass. So we'll see that whenever there would be a beta decay, there would be no change. There would be no change in atomic mass. Then a third kind of decay is gamma particles, and gamma particles, they simply represents an excess energy. Okay, so let's study little bit about the alpha decay. So let's say there is some parent particle, that is uranium, with its atomic mass of 238 and atomic number of 92. Since it is having an unstable nucleus, it decays and emits a alpha particle. As we have already mentioned that alpha particle is like a nucleus of helium atom. So what you'll find is that there would be decrease in atomic mass. Atomic mass reduced by 4. Why 4? Because 2 protons and 2 neutrons. And atomic number, very good, you know, that's really good, reduced by 2. So whenever there is a alpha decay, there is a change in atomic number as well as there is a change in atomic mass. Atomic number reduces by 2 and atomic mass, it reduces by 4. And since the atomic number changes, the identity of an atom will also change. That is, now it will not be regarded as uranium, now it would be regarded as thorium, because atomic number instead of 92 now it is 90. So what has changed? Important thing here in alpha decay, an identity of an atom is also changed. Identity of atom has changed. That is, now the progeny atom is of smaller size, having a lesser atomic number, or atomic number reduces by 2 and atomic mass reduces by 4. Some other thing is that these alpha particles, since it is like a nucleus of an helium atom, so these alpha particles, they are positively charged. They are positively charged. You will find that out of alpha, beta and gamma, these alpha particles, they are having a least energy and least penetration power. That is, even they cannot cross a thick paper sheet or mica or aluminum sheet. They will not able to cross that because they have a lesser energy. But they have, you can say that they have a maximum ionization effect. If you want to note it down, we'll study later on, but they have maximum ionizing effect. Maximum ionizing effect is by the alpha decay. Okay, note these things, these are very important. Let's move further. Then after alpha decay, now let's talk about a beta decay. In beta decay, a simplest beta decay could be a negatron decay, because again beta decay could be of two types, that it could be a negatron type or it could be a positron type. In general, whenever we talk about a beta decay, we talk about a negatron type of beta decay. Whenever there is a negatron type of beta decay, what happens is there is a emission of one electron. This electron is basically a negatron particle with another particle to balance is an anti-neutrino, which neither has a charge nor has a mass. Electron do not have mass, but it has at least charge of -1. And during this negatron type of decay, since the negatron, which is equivalent to electron, do not bear any mass, so the change in mass is 0. So during negatron beta decay, there is no change in atomic mass, but atomic number increases by 1. In other words, identity of atom get changed. Identity of an atom will change. For example, here you are starting with a carbon 14 and you are ending with a nitrogen 14. Since both are having a same atomic mass, so we are saying that atomic mass is not changed. It could be represented like this, that this is a C14 form, and this C14 form, which is unstable because there are 6 protons and there are 8 neutrons. And since neutrons, they are greater as compared to the number of protons, these neutrons, they are making this system, this nucleus, unstable. So this excess neutron split, one of the neutron, so we can say -1, one of the neutron it splits into a negatron particle which bears a negative charge and no mass, and a proton extra neutron. Now it split into a negatron and proton which bear a positive charge and rest of complete mass. So minus plus, these two will become, again neutron will become 0, and no mass and complete mass, it will be a mass equivalent to a neutron. This particle which is without a negative charge and no mass, it is released in form of negatron particle. Although beside this there is one more particle is also released, that is an anti-neutrino, as we have mentioned. Whenever a neutron split, there is also release of anti-neutrino, and whenever a release of, or whenever there is splitting of a proton, a neutrino particle will be emitted. So that you study in a radioactivity. So here there is splitting of a neutron, and whenever there is splitting of neutron, there would be a release of anti-neutrino particle. That's not much important, and that is also not our focus point at present. Now what has happened is, since one neutron has split, so there is decline in the number of neutron. Now the number of neutron which has left behind is 7, and since due to the splitting of one neutron there is an increase in one proton, so number of proton also has become 6 plus 1, that is 7. So now there are 7 protons and then there are 7 neutrons, so mass is 14, but since the proton is 7, now it will not be regarded as carbon, it would be regarded as nitrogen, and obviously identity has changed. So what is benefit? So you can see over here C14, when it get converted to an N14 plus a beta particle, which is, you can write it like this, carbon 6 get converted to nitrogen 7 plus an anti-neutrino particle. The unstable form of C16, it is being converted into a stable form, that is N14, and that is the, we can say, property, or that is the reason why basically there is a radioactive decay. Okay, just note it down. Okay, now you can apply your mind over here. Just I am asking you that if there is, let's say, tritium hydrogen, and if it get beta decay, this is a tritium hydrogen, and if there is a beta decay, what you will form? Just give me an answer. There is a beta decay with mass of 0, charge of -1, with an anti-neutrino particle which do not have a charge or a mass. What you will get? Just apply your mind. Okay, yes, you are right. That is, since the atomic number will remain same, it will remain 3. Sorry, atomic mass will remain same, it will remain 3. But the atomic number will increase by 2, it will become 2. Now instead of hydrogen, this would be referred to as helium. So tritium hydrogen on a radioactive beta decay, it get converted into an helium, which is comparatively stable. as compared to a tritium hydrogen. So C14 with time it get converted into a non-radioactive form referred to as N14. Tritium hydrogen with a time it get converted into a non-radioactive form referred to as helium. Okay. Then similarly, now let's talk about a positron decay. In positron decay often happens when the number of protons they are more as compared to a neutrons. Means there is a disequilibrium, and the reason for a disequilibrium is the number of protons they are more as compared to the number of neutrons. For example, you can consider a case of a O15. You can consider a case of O15. You know that the atomic number of this is 8, so number of proton in this would be 8 and number of neutrons would be 7. So obviously in this case the number of protons they are more as compared to a number of neutrons. So in this case neutron is not going to split. In this case, yes, the extra proton splits. So in positron decay, proton splits into neutron plus positron. Neutron is no charge and entire mass of a proton, while positron is positive charge of a proton but no mass. So it could be represented like this, like an electron with 0 but with a +1 charge, that would be referred to as positron. So in this case, since proton splits, there is a decline in the number of protons. So since there is a splitting of proton, the atomic number reduces by 1, but atomic mass will remain same. Why atomic mass will remain same? Because there is decline in no doubt one proton, but there is a increase in one neutron. So decline in proton, it is being counterbalanced by increase in neutrons. So that's why the mass of atomic mass remains same. That is let's see about this example. This is a O15 radioactive form where there is, out of these, let's say there were 8 protons and then there are 7 neutrons. So protons split by 1, there is an increase in number of neutron by 1. Now what happens is protons become 7 and the neutrons become 8. That is O15 get converted to N15, and N15 no doubt is a heavier isotope of nitrogen but is a non-radioactive form, and the particle which is being emitted is referred to as a positron particle. Are you getting this? What we mean by negatron decay and what we mean by a positron decay. You have to keep only one thing in mind, that in a positron decay there is a splitting of a proton, and in negatron decay there is a splitting of a neutron. In positron decay atomic number reduces by 1, in negatron decay atomic number increases by 1. Even the, we can say, penetration power of the positron decay is far less, it is very less. The positrons we use in one medical approach referred to as positron emission tomography. It's a one medical technique often utilized for identification of the cancers, especially brain tumors, on basis of the utilization of glucose, a radioactive form of glucose. So, positron emission tomography, as its name suggests, it depends upon or rely upon the emission of positron. So, what are positron? Positron are the particles which are similar to electron, only the difference is that their charge, instead of minus, it is a plus. So, we have studied alpha decay. We have also focused on a beta decay, which could be of two types: negatron type of beta decay and a positron type of beta decay. Okay, now moving further, another form of a radioactivity which is comparatively lesser known is referred to as an electron capture. Electron capture, as its name suggests, whenever a nucleus is very positively charged, or basically a excess proton in the nucleus, it capture the electron from a innermost shell. Obviously, whenever a proton will capture an electron, it will convert to a neutron. So, like the positron decay, here also the atomic number is going to reduce by 1 and atomic mass will remain same. So, in electron capture, what happens is the proton of positively charged nucleus captures electron from innermost shell. So proton, since it is capturing an electron, the mass of proton, let's say charge is also +1, this is mass is 0, charge is -1, it get converted into neutron whose mass would be 1, charge would be 0. So you can easily assume that what is going to happen? Yes, in this case of electron capture, atomic number decreases by 1. Why? Because there is a decline in one proton is declining. But the decline in the proton is being compensated by increase in one neutron. So atomic mass will remain same. Atomic mass will remain same. That's why such a phenomenon where a positive charged nucleus is capturing an electron from an innermost shell, this is referred to as electron capture. As an example, we can take a case of a potassium-40, which often is involved in electron capture. So this is the potassium-40 with 19 protons and 21 neutrons. If it's positively charged, nucleus captures one electron. Now there becomes a 18 proton and 22 neutrons. That is there is a decline in one proton, but there is increase in one neutron. The properties would be now like an argon, and argon is comparatively stable. And whenever there is an electron capture, there is an radiations, emission of an energy. Okay, so this phenomenon is referred to as electron capture. Another form of radioactive decay is referred to as gamma decay. So gamma decay is an emission of radiation whose wavelength is approximately 10⁻⁻12 meter. So in gamma decay, basically it is an emission of radiations whose wavelength is approximately 10⁻⁻12 meter. In this case, there is no particle emission, only excess energy is released. That is, the gamma rays has charge of 0 and mass also of 0. That is, let's say there is a decay of technetium. So technetium 99, it undergo a gamma decay. So gamma rays neither has a mass nor it has charge. So identity of a technetium has not been changed. Technetium, it has remained technetium. But what has happened? Happened is it was having with a more energy. Nucleus was unstable because it was having an excess energy, and this excess energy, it has been released in form of radiations. So sometimes the instability in nucleus may be due to an excess energy, and this excess energy could be released in form of gamma rays. Most often what you find is gamma radiation is seen with a beta decay or an alpha decay. For example, if we take a case of radon, which emits alpha particles, and beside alpha particles, excess energy is released in form of gamma rays or gamma radiations. So radon, no doubt it get into polonium, whose mass is reduced by 4 and number is reduced by 2. But with an alpha decay, you are also getting a gamma rays or a gamma decay. So basically, gamma decay, it represents an excess energy. Gamma rays, they have a highest mutagenic potential. They are most energetic. एनर्जी के हिसाब से भी उनकी एनर्जी सबसे ज्यादा है. That is, if you look at the penetration power of these alpha, beta or gamma rays, what you find is that alpha rays, as already I have mentioned, is fail to cross even a thick paper sheet. It do not have much penetration power. Beta decay, having comparatively more energy, can cross a paper sheet but cannot cross a thick aluminum sheet. So any radioisotopes is enveloped in a aluminum sheet and it decays the beta particles. Beta particles will not able to cross that aluminum foil. Neither alpha particles can cross that foil, but gamma radiation can easily cross that aluminum foil. The gamma rays, they are having a maximum penetration power. That is, they can easily cross paper, aluminum, although lead is regarded as one of the good insulator of radiations. But if there is a thin lead sheet, even gamma rays, they can cross this. So if there is a thick sheet of lead, then only it will able to stop the emission of radiations. And that's why often these radioisotopes, they are kept in a thick sheet or a box made of lead, so that these radiation should not escape out of it. We can summarize these three different kind of radiations over here that you have already noted down, that whenever there is an alpha decay, atomic number is declined by 4, atomic number declines by 2. Whenever there is a beta decay, and in a beta decay we are assuming that we are talking about a negatron type of beta decay. Whenever there is a negatron type of beta decay, there is no change in a mass, but atomic number increases by 1. Whenever there is a gamma decay, there is neither change in atomic mass nor there is the change in atomic number. In a positron type of beta decay, although there is no change in mass, but there is a decrease in atomic number by 1. Whenever there is an electron capture, the same thing happens like a positron capture, that is, the atomic mass remain unchanged, but the atomic number reduces by 1. Now, except the beta gamma decay, in most of cases the identity of an atom get altered. So the amount of radioactive form which was available at that particular moment will not remain same after some or certain duration. It will decline, because it may with time a radio active unstable form of an atom, it get converted into a stable form of atom. From here we define one term that is referred to as half-life. So what is half-life? So radioactive half-life, which is referred to as T-half, radioactive half-life is the time duration in which the concentration of a radioactive material reduces to half. So T-half is the time duration in which original radioactive material reduces to half of its concentration. That is the time duration which is being taken for converting 100 gram of radioactive material to 50 gram, or for converting 1 gram into 0.5 gram, or 10 microgram into 5 microgram, or 1 nanogram to 0.5 nanogram. This is constant, referred to as half-life. So T-half, it depends on 0.693 by lambda, where lambda is a disintegration constant. So if there is more disintegration, means there is more disintegration constant, then the material will disintegrate very fast. And if material is disintegrating very fast, means half-life would be lesser. That is, in a very small period of time, its concentration reduced to half. Since the disintegration constant is a constant for radioisotope, and since the disintegration constant and half-life is inverse relationship, so even the half-life will also be a constant. So for example, noted down, the half-life of tritium hydrogen is 12.3 years. The half-life of carbon 14, which is often utilized in radio dating, is around 5,730 years. 32 phosphorus, which is often utilized for radio labeling of nucleic acids, is having a half-life of 14.3 days. Uranium-235, often utilized for radio dating of fossils, having a half-life of around 0.7 billion, or more accurately 7.1 into 10⁸ years. Uranium-238 could also be utilized for radio dating of very old fossils, and its half-life is 4.5 into 10⁹ years. That is, this is the age of Earth. Almost the half-life is equivalent to the age of Earth. So these are the half-life of certain radioisotopes. So as I have already mentioned what we mean by half-life. So half-life is the time duration. For example, we are starting with a cobalt-60, which is having a 10 gram, and after you can say certain duration of time, whatever the radiations which are coming from that, it reduces to 50%. The time duration where the radiations falls to a 50%, that time duration would be referred to as first half-life. Similarly, and in this case, it comes out to be around 5.27 years. Again, in next 5.27 years, that is in second half-life, the amount will convert from 5 gram to 5 gram to 2.5 gram. In next 5 years, it will reduce to 1.25 gram, and so on. Okay, are you getting this? So basically, what would be the amount of radioisotopes which will remain behind? Means out of 10 gram, what amount will remain behind? Whether 5 gram will be there, or 2.5 gram would be there, or 125 gram would be there, or 0.625 gram would be there, or even lesser than that will remain. That depends upon number of half-life. As the number of half-life increases, the amount of radioactive material which is going to remain there of an original will reduces, and we can establish a very simple relationship, that is the isotope remaining equal to half, because we are talking about half-life, half raised to power n, where n is the number of half-life into the starting material. Okay, so let's say there is a 100 gram of C14. This is a original amount. Okay, that is an original amount, and let's say assume that T half of C14 is 5,500 years. This is also given to you. Okay, now what would be isotope remaining after 22,000 years? If we monitor it after 22,000 years, what would be the amount of radioisotopes which is going to left behind? So first you have to find out what are the number of half-lives. What is the value of n? So n is number of half-lives. Total time divided by duration of half-life. Total time is 22,000 years. Half-life is 5,005 years. So you get that there are 4 half-lives, number of half-lives. So isotopes remaining would be half into raised to power 4, and original amount was 100 gram. You can say that 2 multiplied by 2 is 4, multiplied by 2 is 8, and further multiplied by 2 is 16. So you could say 1 by 16 into 100 gram. That is, it would be 1/16th fraction of 100 gram, which would come out approximately 6.125 gram. This much amount of C14 will left behind after 22,000 years if we start with 100 gram. In other words, you can monitor the amount of C14 in any fossil, that what proportion of C14 is there in that fossil as compared to present living organism, and you can establish its that how old that fossil is. For example, if you find some fossil which is having a 25% of C14 as compared to the original, how old is fossil? What you will say that is 25%, means from 100% first it would have become 50%, and then in next half-life it would have been 25. It is having 25% of original, means there are two half-life. There are two half-life means, and every half-life is, if it is 5,500, so in two half-life means it is 11,000 years. So we'll see that the fossil is around 11,000 years old. Okay, so this is isotopes, then radioisotopes, and little bit idea about the half-life. Now coming to the point, what we mean by isobars and isotones. Some simple terms. What are isobars and what are isotones? Just note it down. Isobars have same atomic mass but different atomic number. They are referred to as isobars. Means this is, now you can say that opposite to what you study in isotopes. In isotopes there is same atomic number but variable atomic mass. In isobars there is a same atomic mass but variable atomic number. So there are two different atoms having a same atomic mass. They would be referred to as isobar. For example, C14 and N14. Both are isobars. Are you getting this? C14 and N14. Both are isobar. Okay, you can say that similarly you can use another term, that is a isotone. Isotone, they have the same number of neutrons. They have same number of neutrons but variable atomic mass and atomic number. Obviously, if the number of neutron is same, then there would be variation in number of protons, and if there is variation number of proton, atomic number will also change and atomic mass will also change. So let's say the oxygen 16 and you can say nitrogen 15 and carbon 14. In all these cases, proton here is 8, proton here is 7, proton here is 6, neutron is 8, here is also 8, here is also 8. So number of neutron in all three scenario is same. So such condition would be referred to as isobars. Got this point? Then we have talked about isotopes, isobars, isotones. Now moving back to an atomic mass. We have said that atomic mass of carbon is 12, of nitrogen is 14, of oxygen is 60. But what are actually units of this? Means what? Carbon is 12 gram? No, it cannot be that much heavier. Or it is 12 microgram? No. What are the units for these atomic mass or atomic weight? So at the unit for atomic mass or weight is atomic mass unit. The name of unit is also an atomic mass unit, that is a AMU, or simply referred to as unit, or simply referred to as Dalton. So what you can say that carbon 12 means there is a atomic mass is 12 Dalton, or 12 units, or 12 atomic mass units. Whatever you find more convenient, you can use that term. That the atomic mass of an oxygen is 16 units, or 16 atomic mass units, or a 16 Dalton. Are you getting this? Basically, these atomic mass units, they are relative units. These are the units as compared to the relative, or these are the relative units as compared to the mass of hydrogen atom. So if carbon is 12, what this suggests us? This suggests us is that carbon atom is 12 times heavier as compared to an hydrogen atom. So the benchmark for this relationship is an hydrogen atom. So carbon 12 means if you put a carbon 12 on one side, on another side you have to keep at least 12 hydrogen atom, and when you put a 12 hydrogen atom on this side and one carbon atom on this side, these both, the balance will show are equal, or the weight would be now equal. So what we can say that what is 1 AMU? So 1 AMU or 1 Dalton, what this means to us? So 1 AMU or 1 Dalton is the atomic is equivalent to atomic mass of 1 hydrogen atom, or more accurately we are talking about that form of hydrogen atom which is having only one proton, or equivalent to 1 proton. You can write it like this also. So 1 AMU is equivalent to mass of 1 proton. So we know that what is mass of 1 proton. So we can say that 1 AMU, since it is equivalent to 1 AMU or 1 Dalton is equivalent to mass of proton. So we can put it like this, that 1 AMU, if you want to convert it into absolute unit, it could be 1.67 into 10 raised to minus 24 gram. Okay, so 1 AMU or 1 Dalton is equivalent to 1.67 into 10 raised to minus 12 gram. So 10 raised to power 24 gram. This is an absolute unit. So in relative unit, we can say that 1 AMU or 1 Dalton is equivalent to 1 hydrogen atom. It is equivalent to 1/12 of carbon atom, or it is equivalent to 1/16 of oxygen atom, or we can say it is equivalent to 1/14 of nitrogen atom. That is 1 AMU or 1 Dalton. And if needed, these relative units, they could be converted into an absolute unit by multiplying it with the mass of 1 proton. So what I want to say is, let's apply our mind on this simple question. If human genome is 3 giga base pair, 3 giga base pair means it is 3 into 10 power 9 base pair, let's say. What will the total molecular weight in gram? Means if we put this on a weighing balance, what would be the mass of complete human genome? Given that one base pair is 660 Dalton. Okay, so it is, you can represent as a Dalton, or it could be represented as AMU, or it could be grams per mole. Means these could be the units which could be utilized. So first it is 3 giga base pair. Okay, so 3 giga base pair means 3 into 10 power 9 base pair, and we know that one base pair is equivalent to 660 Dalton. So you can put it like this: 3 into 10 power 9 into 660. It is given one base pair is equal to 660 Dalton. And what we know that 1 Dalton would be equivalent to, yes, 3 into 10 to the power 9 into 660, and 1 Dalton is equivalent to 1.67 into 10 to the minus 24 gram. So now your answer, if you multiply all these things, you will get your answer in gram, that is 3 into 660 into 1.67 10 to the power 9 and 10 to the power 24. You simplify them, you will get 10 to the power 15 gram. Further, it would be like this: 3 into, let's say 6 into 1.67, means instead of 660 you are dividing with 1000, into 10 to the minus 12 gram. And if you multiply 3 into 0.66 into 1.67, you will get approximately value of 1, approximately 1 into 10 to the minus 12 gram. So you can say that it would be 1 picogram. The 10 to the minus 12 is referred to as pico. So in this way, this could be solved. Values of Dalton could be converted into 3 into 0.6 into 1.6. This will approximately come up to 3. So it would be like this, convert to 3 picogram. So the relative units, if needed, could be converted into an absolute unit, and we very rarely convert into an absolute unit. We will never say that a human genome is a 3 picogram. Most often we will use the term that is a 3 giga base pair, or we could put it in Daltons. Very rarely we will need it into you can say absolute units. Okay, so what are the units of an atomic mass? The unit for an atomic mass is an atomic mass unit, or Dalton, or simply referred to as unit. 1 Dalton is basically a mass of 1 proton, and mass of 1 proton is 1.67 into 10 to the power minus 24 gram. So up to now we have talked about the protons and neutrons which resides in nucleus. But we know that beside protons and neutrons, in the surrounding, means beside this is the nucleus, in surrounding there are also electrons. So let's talk little bit about the electrons. Now the electrons which are present around a positively charged nucleus, they are mainly responsible for the reactivity or for the chemical properties. Okay, that is for the bonding. If we say that nucleus is more responsible for identity, then electrons, they would be more responsible for the formation of a different kind of bonds, because we will see that bonds, they would be formed by either sharing or transfer of electrons. So these electrons, you see that first they are present in a shell, and every shell, it has, on basis of energy, it is having a subshell, and in subshells there are orbitals, and in each orbitals there could be a pair of electrons which are antiparallel to each other. So what are the, so first what we are getting is the shell. Shells, basically these are the shells. It includes the orbitals with same principal quantum number. For example, 1s, this would be a 1 shell. Similarly, another shell could be 2s, 2p, because they both have same principal quantum number, they both belong to a same cell. Similarly, 3s, 3p, 3d, these would be another shell. So in shell, this is shell number 1, this is shell 2, this is shell 3. So the shells is a combination of variety of orbitals, and these orbitals, they are having a similar type of principal quantum number. Then within a cells there are subshells. So these 2s, 3s, 3p, these 3s or 3p or 3d, these would be referred to as subcells. Okay. Then within a subshell, for example, in a 3s, there is one orbital. in a 3p you will have a 3 orbitals like this. You will control, and maximum there could be a 6 electron, and in 3d there could be a 5 orbitals. So within a shell there are subshells. That is, in a shell 2, 2s and 2p are subshell. In 2s there is 1 orbital. In 2p there are 3 orbitals, and every orbitals can have maximum 2 electrons. Okay, you are getting this point? So in this way the electrons, they are arranged around the nucleus. First there is shell, then there is a subshell, then there is an orbital, and in orbital you will find the electron. Okay, then what could be the maximum number of electron in a shell? That depends on shell number. A simple formula could be applied, that maximum number of electron in shell would be equivalent to 2n square, where n is number of shell, or we can say it is a principal quantum number. So for example, if we are talking about the shell K whose n number is 1, it has designated by K, it has a subshell that is S, has single subshell S. Number of electrons in this, because principal quantum number is 1, it will be 2 into 1 square, that is 2. Similarly, another second shell is 2, represented by L. Subshells over here is S and P. Number of electrons could be 2 into 2 square, that is 8, and so on. In shell number 3, represented by M, S, P, D, 2 into 3 square, that is you can get 18 electrons. In N is equal to 4, represented by N will have subcells S, P, D and F. 2 into 4 square, that is 32. And S has 2 electrons. P can maximum has 6 electrons, that you have studied in chemistry. D can maximally have 10 electrons, and F could have 14 electrons. Why so? Because in S there are number of orbitals. If you talk about the orbitals, there is only 1 orbitals, there are 3 orbitals, there are 5 orbitals, there are 7 orbitals, and every orbitals can have a 2 electrons. The same thing could be represented in this table. That is, shell number 1 is K, 2 is L, M is 3 and N is 4. Subshell, it could be S, which could have a 2 electron. In a shell L there are 2 subshells, namely S and P. S could have 2 electron, P has 6 electrons, that's why there are total 8 electrons. M, it is shell 3, will have 3 subshells S, P, D. S could have 2 electron, P could have 6 and D could have 10, so total 18 electrons are possible. Subshell 4N will have S, P, D and F. S will have 2 electron, P will have 6 electron, D will have 10 electron and F will have 14 electrons. So total 32 electrons are possible in F subshell. Okay, so this is how the electrons, they are filled around the positively charged nucleus. Now if you look the distribution of electrons, often it is observed that first the inner shell they are filled and then the outer shells they are filled. For example, in hydrogen atom only there is a 1 electron, although blue dot represents it is partially filled, but the first electron is filled in a K. Once a K shell is filled, then only the second electron shell is filled. Once this second shell is filled, then the third shell is filled. Now basically, as I have mentioned, that most of the chemical properties, they are basically due to the number of electrons in a shell. So just concentrate for last these 10 minutes, then we are going to sum up this topic. Okay, just have a patience of 5 minutes. That whenever the electrons, or more accurately we can say whenever the shells are completely filled, the atoms are stable, and such atoms, they are non-reactive. Such atoms, they are non-reactive. That is, they do not react or they do not interact with other atoms. For example, most of the inert gases like helium, neon, etc. You can see here it is helium is having full first and second shell. Neon, it is having a completely filled the first and second shell. Argon, it has completely filled first, second, third, and so on. Whereas the carbon, nitrogen, oxygen, or sodium, magnesium, phosphorus, chlorine, or hydrogen atoms, they do not have a completely filled shells. And if they do not have completely filled shell, they would be reactive. So what we want to say is that atoms with partially filled shells always try to lose, gain, or share electrons so as to achieve noble gas configuration, or so as to achieve stability. And such atoms are referred to as reactive atoms. ऐसे atoms को क्या, such atoms, these would be referred to as reactive atoms. Okay, that is, such atoms, they will try to interact with similar kind of atoms or with a dissimilar kind of atoms. Purpose is to achieve the stability. So atoms, these could be reactive or atoms could be non-reactive. Non-reactive or inert atoms are those which have a completely filled electron shells. So there is no need to lose, gain, or transfer electrons. While the reactive atoms are those which has partially filled electron shells. So they will always we can say always they will keep on looking for the gain, transfer, or sharing of electrons so as to achieve the configuration similar to a noble gas, so that they can also become stable. Now, when we talk about the electrons, we define one more term, and that is referred to as an electronegativity. Basically, electronegativity is an affinity. It represents an affinity for electron. That is how eager an atom is to attract a shared pair of electrons. So even we can say that is affinity for an electron, or it is a tendency to attract shared pair of electron. If you look the trends of the electronegativity of atom, what you find is in periodic table, as you move from the left to right, electronegativity increases. So there is an increase in electronegativity. That is, the atoms like lithium or sodium or magnesium, they instead of attracting electron, they try to lose electron. They are not electronegative, they are positive. They are often electropositive. And the atoms like on this side, like fluorine, chlorine, or sulfur, oxygen, these are electronegative. So from electropositive we are moving to an electronegative. And as you move from top to bottom in different rows, again there is a decline or there is a decrease in electronegativity. As you move from top to down, the electronegativity decreases, because the electrons basically they are attracted by nucleus, and as atomic size will increase, the electrons they will move far away from the nucleus. The tendency to attract an electron will decrease. If the electrons they are in outermost shells, instead of attracting these electrons, they can be easily knocked out or they can be easily lost by that particular atom. But since from periodic table point of view, the most electronegative would be fluorine and least would be francium. So electronegativity could range from fluorine to francium, but neither fluorine nor francium it is present in biological system. If we talk about those atoms which are present in biological system, what we find is the maximum. So here we are talking about order of electronegativity. So order of electronegativity for atoms in biological system, what you find is the most electronegative atom, yes, you know, is the adjacent neighbor to the fluorine, that is oxygen. After oxygen there is the nitrogen. After nitrogen, sulfur and carbon, they have almost similar electronegativity. Then comes the phosphorus and hydrogen, again they are having a almost similar electronegativity. So most electronegative, in other words, is the oxygen. We are talking about the 6 atoms, that is oxygen, carbon, hydrogen, nitrogen, phosphorus, sulfur. What you find is that the maximum electronegativity is oxygen and the least is of the hydrogen or a phosphorus atom. Okay, so this is the concept of electronegativity. So my dear friends, up to now what we have studied is about the atoms, about the isotopes, about the isobars, about the isotopes. Now we also have some information about what we mean by electronegativity. In next session, now we are ready to study what we mean by bonds, because already, as I have introduced, that if the shells they are not completely filled, atom it will always keep on looking for electrons from somewhere. It will interact with another atom, and when two atoms they start interacting, we will say that now they have been bonded. So interactions are simply bonds. Okay, so thank you. Hopefully you would have liked this session. Try to make again short notes. Try to be conceptually correct. Okay, so, and if you have any doubt, you can just post it in the chat box. Thank you.